Good day, panelists. We are the researchers of the study MIMOSA pudica, Makahiya or shy plant, as an antimicrobial spray for wound. I am Angel Kate H. Vivas.
I am Kirsten Kibayani.
I am Kristen Calyon.
I'm Trisha Reyes.
And we are from STEM 2C.
Next slide, please. Let us begin with the summary of revisions based on the panelists' recommendations. The panelist has given 5 recommendations in total in order for the improvement of the whole study. 3 out of 5 of these recommendations were implemented, and I will discuss them one by one. First, the researchers were advised to consider the study of using Makahiya as an antimicrobial medication product for scabies or gullies in the form of spray rather than a component of toothpaste or mouth cleanser or mouthwash. Instead of scabies, the researchers tested it on wounds. This revision may be viewed under the data gathering. Next slide, please. Secondly, the researchers were told to look for a subject of testing. They suggested to conduct to children and to make sure to have secure consent from parents or guardians before the testing of the product. Though there were numerous children near the researchers who were willing to be tested, but it was advised by the research advisor, Sir Alvin Barcelona, to not have children as test subjects, as they are unable to provide consent despite being willing to participate. A parent's consent is a must. Next slide, please. Third, the researchers were told that they need to prepare different formulations of Makahiya. The revision can be viewed in the data gathering. Next. Fourth, the researchers are advised to add more related literature on the chemical composition, characteristics, and nature of Makahiya as an antimicrobial. The revision can be viewed on the second chapter of the paper. The fifth recommendation given by the panelists is to look into an area where Makahiya is abundant. Instead of measuring Makahiya leaves by counting the numbers of leaves for testing, measure the leaves in grams. Instead of counting the number of Makahiya or shy plant leaves, the researchers changed the method and measured the plants used in grams. This can be found in the data gathering. Next slide, please. For the statement of the problem of this study, firstly, the researchers wanted to know how effective the MIMOSA pudica antimicrobial wound spray is in terms of abrasion wounds, disinfection of dirt and bacteria, and coverage. Second, the researchers wanted to know which antimicrobial spray seems to be most effective based on the 2 specific concentrations of MIMOSA pudica. Lastly, the researchers wanted to identify the effects of the proposed antimicrobial.
Next slide, please. Let's proceed to the research design. The study is an experimental study which focuses on examining the potential of MIMOSA pudica as wound healing spray. Its goal is to investigate if Makahiya leaves would be effective and feasible if used as wound spray. This study focused on the effect of wound healing activity on changes of wound when the right amount and exact concentrations were applied. The researchers have observed the capability of Makahiya leaves to be an effective wound spray that, based on the relevant studies and literature that have been gathered. Next slide, please. Moving to the next part, which is the data gathering procedure. Firstly, all the ingredients and equipment were prepared. Second, creating the MIMOSA pudica, Makahiya or shy plant spray. Third is the researchers observed and documented the before and the after trials of the two concentrations, the 100 and 200 grams of Makahiya leaves. And lastly, the results were gathered and summarized by the researchers.
Next slide, please. The next part would be the findings that the researchers have analyzed after applying the antimicrobial spray with different concentrations to the wounds. We've found that wounds sprayed with the antimicrobial wound healing spray dried easily after 3 days. By comparing the wounds that were treated with the 200 milliliters concentrations and 100 milliliters concentration, it was observed that the wound with the 200 ml formed scabs earlier than the 100 ml concentration. The scab formed on the third day of treatment with the 200 ml concentration, while the scab formed on the fourth day with the 100 ml concentration. The peeling also started on the fifth day of documentation for the 200 ml concentration, while the scab only fully developed for the wound with the 100 ml concentration. However, the redness darkened for the wounds with 200 ml on the fifth day, while the redness disappeared for the wounds with 100 ml concentration. Besides, the scratching caused by the itchiness from the first application of the 100 ml concentration, there were no significant changes in sizes during the documentation. Next slide, please. These were the proof before application. Next slide. And these were the proof after application. Next slide.
And now, in the light of the findings, the following conclusions were derived. MIMOSA pudica, or so-called Makahiya, played a vital role in the reduction of the inflammation and scars of the wound area. Mimosa pudica, as observed in the study, has the innate ability to heal wounds even on its own, and it has been proven by the other studies that can be found in the chapter 2 of this research document that it is possible that the wound healing ingredient. Then, second is, Makahiya helped amplify the healing of the skin of the respondents because its chemicals promote cell and tissue growth. The ones used and observed in this study also have antimicrobial properties and the plant's ability to prevent scarring and continuous application. Third, the made product from Mimosa pudica showed a satisfying result that enables it as an antimicrobial plant or a product in the market. Through the study and observations and outcomes also, the researchers concluded that the product is feasible as an alternative wound healing spray at the market and for some home use for those that in need. And lastly, as the findings showed, the researchers found the perfect concentration of 200 ml of the Mimosa pudica to the spray as the wound heals faster and the scars fades quickly compared to the 100 ml concentration.
Let us now move on to the recommendations. After finishing our research, we discovered areas where future researchers could improve their studies. First is by comparing the wound healing spray to other skin healing and cleansing solutions to see how they compare. For example, our Dermaplast First Aid Spray, Qtacept Hospital Grade Woods, Antiseptic, and etc. Second, to further test the effect of the spray, consider using the antimicrobial spray on the moderate or superficial skin damage that isn't confined to abrasions. For example, animal bites, scrapes, and burns. For our third, the amount and the concentration of each ingredient must suffice for the wounds. consider making more concentrations to differentiate and find out the best antimicrobial food spray. And for the last is to consider laboratory testing of the spray to determine the efficacy of its antimicrobial and healing process of the wounds.
In order to create the product, the ingredients and equipment used are Makahiya plant, strainer, olive oil, spray bottle. Next slide. We also use blender, measuring cup and syringe, funnel, and pitcher. Next slide. For the process of the final product, the ingredients and equipment are prepared. After the preparation of all the things needed, the Makahiya leaves were then cleaned and boiled with oil, then later strained. After that, all the ingredients are blended together. Next. Then it is poured inside the spray bottle to be used by the participants of the study. That is all. Thank you for listening.